Welcome. Analytical Method Validation or AMV. AMV documents are necessarily reviewed by regulatory agencies during regulatory inspections. Different regulatory agencies having their own EMV guidelines. Company perform EMV as per its own defined procedure because it is impossible for any company to perform different EMV for different authorities as per their guidelines. That is the reason comments are given on AMB during regulatory inspections and deficiency letters are given during scientific review. Now question is what is its solution? I have very good experience in handling such issues from different authorities. That is why I decided to share my skill based knowledge on this topic. I am going to cover this topic in multiple sessions. This is the first session, and in this session, I am going to discuss what is analytical method validation or AMV. In this session, you will learn what is AMV, what has to be validated, what are different guidelines on AMV, what type of method requires AMV, what should be a strategy for AMV. Now question is what is validation and what has to be validated? Establishing documented evidence which provides a high degree of assurance that a specific process will consistently produce a product meeting its predetermined specifications and quality attributes. AMB also tells about sensitivity and limitations of the method which helps in avoiding any error while performing the analysis. Validation includes both equipment and methods. There are several guidelines on method validation. Some of the guidelines are the International Conference on Harmonization (ICH), the United States Environmental Protection Agency (US EPA), the American Association of Official Analytical Chemists (AOAC), the United States Food and Drug Administration (US FDA), the United States Pharmacopoeia (USP). Following methods are validated: HPLC, high pressure liquid chromatography; GC, that is a gas chromatography; TLC, that is a thin layer chromatography; titration, that is a chemical titration method. We validate methods for final API, intermediates, raw materials, cleaning of equipments. Adopt following strategy for method validation. Perform pre-validation experiments before start the validation activities. Develop a protocol or operating procedure for the validation. Define the application, purpose, and scope of the method. Define the performance parameters and acceptance criteria. Define validation experiment. Verify relevant characteristic of equipment. Define materials, for example, standards, marker, and reagents. Perform full internal and external validation experiment. Develop SOPs for executing the method in routine. Define criteria for revalidation. Define type and frequency of system suitability tests. Document validation experiments and results in validation report. Analytical methods. The chromatography analytical methods are divided into method versus external standard of the tested analyte, which includes assay, residual solvent, and some of the impurity profile methods. Method versus external standard different from the tested analyte. Most of the impurity profile method. Methods of area percent or area normalization. Purity, which includes solvents, reagents, and some of the raw materials like key starting material or starting material, impurity profile for starting material and intermediate. Methods of impurities by area percent or area normalization for final material. That is all about this session. I would like to request you to write at least one of your learnings in the comment section. If you are new on this channel, then subscribe the channel and give a like to the video. Let us see you soon in the next session. Thank you for watching this lecture.